Good evening, everyone. Today is your week 8 session. So, in this session, we'll be talking about the microscopy, some functional details of cryo EM, and along with that, we'll look into the detail of cryo electron microscopy, in which we'll see that how the data is collected and how it is analyzed, and then we'll see that how it has advanced through years. And then we'll look into few databases which stores the data, structural data information about the proteins, nucleic acids, and all the databases which are available and what is their current advancement. So all those things we'll look into. Then we'll practice few MCQs, and along with these topics we'll also look into the current literature, and then I'll take your doubts. So let's start with the microscopy. So as we all know that our human eye, it cannot see objects which are smaller than 100 micrometer in diameters. So for that, in some cases we use hand lenses which have the magnifying glass which provides 2 to 10 times magnification, and these are the simplest microscopes. Then comes light microscope, which uses the visible light and the system of lenses to magnify small samples. So for example, if we have a small sample, then with the help of magnifying lens or with the help of lenses, we can enlarge the image. So our eye will see an enlarged representation of a small image or small object. So for that reason we use light microscope. So in the light microscope we uses visible light as a source of illumination, whereas if we talk about electron microscope, then instead of visible light we use beam of electrons to achieve much higher magnification and resolution, which allows the visualization of structures as small as 0.05 nanometers. So that is the main principle, that if we want to see the very smallest structures, then we go for electron microscopy instead of light microscope. And in the later slides we'll see that why do we use electron microscopy. So this is the representation that with the naked eyes we can see up to 100 micrometer of structures, whereas with the help of light microscope we can see objects from ranging from 1 millimeter to 100 nanometers. Whereas if we want to see the objects which are even smaller than 100 nanometers, then we go for electron microscopy. Yeah, this is also the another representation which talks about that as the size decreases, we move from macroscopic to microscopic and then to particulate objects. So the particulate objects are the objects which are smaller than these 100 nanometers. So they comes under particulate matters such as viruses, protein molecules, water molecules, atoms. So for these kind of objects or particulate matter, we need electron microscope, and specifically under electron microscope we need scanning tunneling microscope, so which is another specialization of electron microscope. So now we'll talk about the compound microscope, or which is the light microscope and its aberrations, that why do we have to move towards the other advancement of the compound microscopes. So compound microscope, it uses an objective lens which is close to the object to collect the light and form a real image inside the microscope, and then this image is magnified by the eyepiece lens, producing an enlarged virtual image for the viewer. So in compound microscope we have two type of lens systems. First one is objective, which is near to the object or the specimen, and it will form a real image before the eyepiece lens. Now this eyepiece lens will collect these rays coming from the objective lens, and then when the light will pass from eyepiece to our eye, then an eye will see an enlarged image which will be a virtual image of the object. So in this way this will magnify the image even greater than that with a single piece of lens. So in the previous slide where we looked into this magnification, in this case only one lens system was used, but in the case of compound microscope we have two lens system. So with the help of two lens systems we can enlarge the image multiple times depending on their magnification powers. So if we have a perfect lens, then all the light rays from a given point of light means all the light rays which are coming or which are passing through this lens, they will converge at a single point which is in the plane of focus and which will form a exact image. So this is in the case of perfect lens, but in compound microscope we face certain aberrations such as spherical aberrations. So what generally happen is that light rays, when they hit the surface or the periphery of this lens, then they get refracted more than those hitting the center part or some middle part. So in this way, the refraction angles are varying, due to which they will not meet at a single point, which they would have met if it was a perfect lens. So in this case, they are meeting at different points, due to which it will form an image which will be blurred image. So this type of aberration is known as spherical aberration, because it is happening near the periphery of the lens system. So that's why it is known as spherical aberration. Then second aberration is chromatic aberrations. So as the name suggests, chromatic, that means it has something to do with the colors. So as we all know that the white light splits into 7 different colors. So when it passes from a simple lens, then it is refracted differently. That means there are different angles at which it gets refracted, and it splits into different characteristic wavelength light. So in this way, the blue light, it will focus shorter, whereas red light, it will meet at some point farther from this blue light. So in this way, they have different focal points at which they meet, so due to which it will lead to an unsharp image with color fringes. So this type of aberration is known as chromatic aberrations. So now these are the problems with the lenses. So we need to have certain modifications done to deal with these challenges. So some advancements are confocal microscopy. So it is an alternative to X-ray crystallography and NMR spectroscopy for macromolecular structure determination. So in the previous session, when we were talking about the Raman spectroscopy, so in that we discussed that in the application of Raman spectroscopy, which was Raman microscopy, the microscope which was used was the confocal microscope. So similarly, this is an alternative, as it eliminates the need for crystallization, and it uses a pinhole aperture to remove out of focus lights, and in this way it enhances the image clarity. So in this way, as you can see, there is a pinhole which only allows the light which are in focus. In this way, it removes the out of focus light, due to which the image clarity is enhanced. So that's why we go for the confocal microscopy, as it can deal with the challenge of chromatic as well as spherical aberrations. Then comes cryogenic electron microscopy, which in short term often called as cryo-EM. So it is an electron microscopy technique applied to samples which are cooled to cryogenic temperatures. So when we talk about electron microscopy, this is a type of microscopy in which our source of light are the electrons. So instead of using visible light, we go for electrons. And then cryogenic EM is a specialized type of electron microscopy. As the name suggests, cryogenic. Cryo means that something which is at very low temperature, at a cold environment. So in this way, if the samples are cooled to cryogenic temperatures, that's why it is known as cryo-EM. And in this, samples are embedded in the vitrified water to preserve their native structure. It involves plunge freezing the sample on a grid mesh using a liquid ethane or propane mixtures. So these all things, that how these samples are prepared and what is the need for using the vitrification or plunge freezing. So all these techniques we'll look in the subsequent slides. So recent advancements in detector technology and software enable near atomic resolution of biomolecular structures. So with the help of cryogenic EM, we can look at the atomic resolution of biomolecular structures such as proteins, nucleic acids. So all for those reasons we use cryo-EM, in which we have a sample, we put that sample into a grid which will hold this sample, and then we freeze the sample so that we can maintain it in its native state. We can freeze it, because in cryo-EM we have to maintain the vacuum environment so that light molecules or other atoms do not interfere with our electrons, or it does not quench our electrons. So in that way, we need the sample which should be in a proper environment in its native state, but without causing any harm to the electrons or any disturbance in the signals. So for that reason, we freeze them, and then we acquire the data after passing the electrons through the sample, and then we click pictures from at different angles. We take the 2D projections, we combine them, we align them, average them, and make a 3D map. And later on, we perform some Fourier transformations to convert it into an electron density map and construct a 3D model. So all these steps which you have seen so far, we will go into them one by one and we'll see them in detail. So don't worry, and by the end of this session, you'll be clear with all these topics. So now let's move to next slide. So before talking further, we must clear certain concepts related to microscopy. So one such concept is resolving power. So resolving power is the ability of an objective lens to distinctly separate two adjacent points. So as in this case, you can see you are only able to see a single point. So this is not resolved. Whereas in this case, you can see that there are two adjacent points, but you cannot distinguish them. They still appear so close together, so they are just resolved. And in this case, these 2 points can be clearly seen as separate points, so this is known as well resolved. So this is the resolving power, which means that how much ability a particular microscope have to see these two objects as separate entities. So that is resolving power. And one more concept which I want to tell you is about the resolution. So resolution means that what is the distance between these two objects, means how far you see them from one another. So that is known as resolution. So if resolution increases, the value of resolution uh, increases, so which means that the resolving power is reducing. So they have some inverse relationship. So resolving power formula is lambda by 2Na, where lambda is the wavelength of light and Na is the numerical aperture. And now we'll see that what is numerical aperture. So numerical aperture defines the range of angles over which a system can accept or emit light. So it is given by Na, and which is equals to n sine theta, where n is the refractive index of the medium and theta is half the angle of maximum light cone entering or exiting the lens. So in this case, this parallel beam of light is entering, and now it is exiting and making a cone-like structure. So this is a cone which is formed. So this theta, it is the half angle of maximum light of cone. So this is the maximum light of cone, and this is the half the angle which passes from the center of the lens to the focal point. So this half angle is known as theta, and n is the refractive index. So if we talk about air, then in air has a refractive index of 1, and then this is the half the angle. So for example, when the light is passing from here, so in this case also we'll take half the angle, which will be theta 2. So here is an example. So this shows that there is an inverse relationship between the numerical aperture and working distance. So this is, if imagine this is your surface. So if from here the means distance at which the lens is placed, if it is greater, then in that case your numerical aperture will be low and theta is 7 degrees, so angle is also less. Whereas in this case, if this distance is reduced, so theta increases, which is 20 degrees. So in this way, if there is increase in theta, so value of numerical aperture also increases. And now in this case, the distance is very less as compared to our primitive examples. So in this case, theta further increases to 60 degrees and numerical aperture is highest in this case, which is 0.87. So in this case, you can see that there is an inverse relationship between the working distance and numerical aperture. So as the working distance decreases, numerical aperture increases, because the theta angle is also increasing with the means shortening of the distance or working distance. So this is about the numerical aperture. Now we'll talk about magnification. So magnification is also an ability of the microscope to see an object as an enlarged image without increasing its physical size. So in this case, as you can see, this is the leaf. This is the actual leaf which is being observed. And now when a person is looking with the help of a lens, then this image is appearing as a large enlarged image. So this image is a virtual image. This is not real. But still you can see a larger picture of the object which is actually present. So this is known as magnification. And the power with which tells us how much an object can be magnified is known as magnifying power. So each lens have a particular magnifying power, and in compound microscope, as we know, there are two types of lens systems which are used, objective piece and eyepiece. So in this way, depending on the respective magnification power of both of them, that will determine that what will be the overall magnifying power of that particular microscope. So this is an example which tells about the magnification versus resolving power. So in this case, it is low magnification and low resolving power, because we do not see these two entities as separate entities. We cannot classify them. So these are appearing as a blurred image. Whereas if we see here, so this is a high magnification of image in which it is magnified, but still the resolving power is low. we cannot still distinguish among these different entities. So in this case, it is a low magnification because picture is small, but it has high resolving power as compared to our previous example, because we can clearly distinctively see them. And in this case, it has high magnification and high resolving power. So its size is enlarged, and we can also clearly distinguish each and every entity which is present. So this is what we mean by magnification and resolving power. So these are two separate terms. Now we will talk about two different types of microscopic techniques. So first one is bright field and second is dark field. So as the name suggests, bright field, which means it has the bright background and specimens appear as a dark objects. So this is the example where the background is white and the specimen is colored, or you can say it appears as a dark. So it is suitable for stained specimens or those with the natural contrast where you can distinguish them. Whereas if they are not stained, they cannot be stained, or they do not have some natural contrast, then you won't be able to see them clearly. So for that case, we go for dark field microscopy. So bright field microscope can be converted into dark field microscope, and the one thing which we need to add to bright field is the stop disc, which is added before the condenser lens. So in case of bright field, the light passes through condenser, it is focused on a specimen, and then from specimen it is collected by the objective lens and ultimately by the eyepiece, and you can see the image. Whereas in case of dark field, you add a central aperture, or which is also known as stop disc, before the condenser. So now the light, it is passing through the periphery of this condenser lens and it is focusing on the specimen. So in this case, light can only pass through the specimen, and the rest of the background light is not able to pass through it. So the background appears dark in this case and specimen is illuminated. So specimen appears bright object. So stop disc blocks the direct light, allowing only the reflected or scattered light to reach the objective lens, and the specimen appears bright against a dark background, and it is generally used for the unstained live specimens. Because as in bright field, when we use staining, so staining can also damage our cells. So in that case, we cannot go for bright field. So in such scenarios, when we have the live specimens, then we can go for dark field microscopy. But there are few drawbacks of this light microscopy, so that's why we have moved to electron microscopy. Now let's see what are those limitations. So light passing through small slits diffracts, forming an interference patterns. So what generally happens, that in a specimen which we are observing, it is a possibility that there are some empty spaces between the cells or between different compartments which are present inside the specimen. So there are some empty spaces from where the light can get scattered, or you can say the light can get diffracted. So after this diffraction, if it passes from objective lens and undergoes a constructive interference, then you can see a proper well resolved image. But if there is destructive interference, then at that part you will see a blurred image. You won't be able to make sense of that particular image. So in this case, and another problem is that if you reduce the slit width, then it will further increase the diffraction, which will make it difficult for the objective lens to collect light properly. So in this case, slit means the distance between the units present inside the specimen. So if that particular width is increasing, then diffraction will further increase. If that width will decrease, then it will increase the diffraction. So in this way, the blurred image are produced because of the diffraction which is taking place. So we have to move to electron microscopy, because the main concept behind electron microscopy is that these electrons, they have higher mass, which is about 9.11 into 10 raised to power minus 31 kg, and it is very heavy than photons. So in light, we have packets which are known as photons, which have mass of about 4.71 into 10 raised to power minus 38 kgs, which is very less than electron, which gives the electron much shorter wavelength and higher resolution in the microscopy. So electrons will have short wavelength than the visible light, as described by the de Broglie equation, which is lambda is equals to h by mv, where lambda is the wavelength, h is the Planck's constant, m is the mass, and v is the speed of light. So in this case, as the mass will increase, wavelength will decrease. So electrons have heavy mass, so that's why they have shorter wavelengths. And if they have short wavelengths, then that means they can even visualize the smaller particulate matter as well. So shorter wavelength allows for the higher resolution, which enable the visualization of smaller structures like viruses, DNA, atoms. So as we have also discussed in few previous slides, where we have seen that means electron microscopy can see very small objects, such as particulate matter, which were not previously visualized by using the light microscopy. So these are the advantages that why we shifted to electron microscopy. So there are two different types of electron microscopes which are majorly discussed. One is TEM and another one is SEM. So TEM is transmission electron microscopy, and from their name only you can distinguish them, both of them. So first of all, electron microscopy uses the accelerated electron beams instead of light to achieve much higher magnification and resolution. And instead of using the objective lens or the eyepiece, they uses instead of using those glass lenses, they use electromagnetic lenses to focus the electrons and enabling the visualization of structures at nanometer and even at atomic scales. So when we talk about transmission, that means something which is transmitting. So in this case, we have an electron beam source. So for that, we have an electron gun, which is typically made with the tungsten filament cathode, and it generates the electron beam. So as the source of illumination are electrons, so for that reason we use electron gun. Now second step is acceleration and focusing. So this beam is accelerated by anode at plus 100 kilovolt and focused using the electrostatic and electromagnetic lenses. So in this case we have anode, which will take the electrons coming from cathode, and due to this attraction for those electrons, the anode will attract those electrons and it will generate the high field, and it will pass it through the condenser lens, and then it will focus them on the sample. So as it is a transmission, so this will allow the transmission of those electrons through the specimen. And then objective lens will, so these objective lenses are also electromagnetic lenses which will bend this electron beam, and ultimately we have objective aperture, which will only allow specific in plane focused electron beam to pass through it, and rest other out of focus beams will not be allowed to pass. And then there are some intermediate lens which are further involved in increasing the you can say resolution of this electron beam, and ultimately there are projector lenses which will collect this electron beam, and in the fluorescent screen the image will be generated. So for this acceleration and focusing, we use this electromagnetic lens systems. And then specimen interaction. So these transmitted electron beam, they carry structural information about the specimen, and magnification and imaging. So beam is magnified using the objective and projection lenses, and the final image is projected onto the fluorescent screen. And if we talk about the resolution, then there are some chances of spherical aberrations in transmission electron microscopy. So for that reason, we can go for high resolution TEM, which can achieve sub-angstrom magnifications above 50 million times. And there are certain disadvantages of this TEM. That means it requires extremely thin specimen sections, typically about 100 nanometers thick. So the main reason that why we cannot work with thick specimen is that because these electrons, they have heavy mass, they are heavier, so they cannot pass if the sample is thick. They will not be able to pass through the specimen. So in that case, the transmission will be quite difficult. So for that reason, we need to have the samples which are very thin for the proper transmission of electrons. So this is how the transmission electron microscope looks like, and these are some of the images taken from the transmission electron microscope. So as you can clearly see that you can see the inner details of particular cells, viruses, and this is specifically of the SARS-CoV-2. So this is the electron micrograph. The pictures which are taken from the microscope, they are often referred to as the micrograph. So this is for SARS-CoV-2. And now we will talk about the scanning electron microscopy. So in this also it's clear from the name that scanning. So that means it will scan the specimen instead of transmitting through it. So scanning electron microscope, it uses the focused beam of electrons to scan the surface of a sample, generating high resolution images based on the electron interactions. So how the electron is interacting with the specimen. So depending on that, it will make the surface pictures of that particular sample. So in this steps are the electron beam generation, for which there is an electron source which will accelerate the electrons and focus it on the specimen passing through the different lens systems. Then this beam scanning, so its scan coil above the objective lens move the beam across the sample collecting surface details. So in this case we have the beam scanner, and then comes the electron interaction. So beam interaction produces secondary electrons. Secondary electrons are the ones, so as soon as this electron beam hit the sample, so the sample will also have certain electrons because it is also an atom. So when this electron beam hits the sample and the another electron of the sample, when it gets excited, so that is known as secondary electron, which will be captured by the secondary electron detector. And there will also be some x-rays which will get generated, so they are collected by the X-ray detector, and the ultimately the other means the other beams which get diffracted, they are further collected by the backscattered electron detectors. So these are the electrons which get scattered back. So these are the some of the electronic interactions which take place. So these are generally captured, and then ultimately the image is formed. So then this is passed through the amplifier, which will amplify all the signals and scan generators, and ultimately image will be formed on a computer screen. So imaging and resolution, signals from the detectors are processed, displayed on a computer screen. Full-sized SEMs achieve the resolution of 1 to 20 nanometers, while the desktop models provide 20 nanometers or more. So these are the resolutions, that how well you can see, how you can distinguish among different compartments present inside the specimen. So we aim for achieving the better resolution. So this is how the scanning electron microscope appears, and these are some of the pictures taken from the scanning electron microscope. So you can see this tells you about the 3D representation of the surface of the specimen. You cannot see what is inside, but you can see what is at the surface. And this is how you can see from the light microscope. This is also one of the image, that if you focus, then how you can distinguish the internal compartments present inside the specimen or the cell. This is from the scanning electron microscope. So in this case also you can scan the top surface of the cell without looking into the inner details of a particular cell or specimen. Whereas this is collected from the TEM, which is transmission electron microscope, where you can focus into the inner compartments of an object, because electrons are transmitted through the object. So this is how different images appear from different types of microscopes. Now if we talk about the cryo electron microscopy, so this is the technique comes under electron microscopy. So cryo means when you are observing the sample at cryogenic temperature, so that is known as cryo-EM. Challenges in studying the biological materials. So we all know that electron microscope uses the electron beams, and this has to be done in a vacuum where there is no air, because air has oxygen and oxygen act as a quencher. So in that scenarios, we cannot allow the other disturbances from the atmosphere or surface to take place during the electron microscopy. So when we use the samples, so in electron microscope, as we know that the proteins or other biomolecules, they are present in the liquid environment where the water is all around. So when we use cryo-EM, then this water is generally evaporated, and due to surface tension, the water will get evaporated, and the biomolecule which is present, it will structurally collapse. So that is the limitation. So means because we need to maintain the vacuum condition, so we have to do this. So due to which there will be destruction of these organic cells, because there are intense electronic bombardments, and then there is also difficulty in preserving the water in biological samples within the vacuum chamber. And then biological materials, they had low image contrast as the high energy electron passed through the specimen. So these are some of the challenges. So how to deal with these challenges? So this is also one of the representation. So in this case there are two images which are shown. So this image A, it shows that when you are using the electrons at 15 electron at 15 electron upon angstrom square, so this is this CuPCCl16 structure, which remains stable with well-defined regions of clean graphene, which are represented as dark spots, and CuPCCl16, which appears as gray, and contamination appears as this bright spots. So this is at this electron volt power which is used, whereas if this electron beam which is passing through the sample, if it is 500 electron upon angstrom square, so means this is, if this is the voltage, then in such cases CuPCCl16 crystals, they become unstable, showing significant structural degradation and dynamic changes in the gray areas, and you can see this white area is enhanced, which is the contamination. So this demonstrates the challenges in SEM and TEM imaging of the biological and sensitive materials, where high electron doses can induce damage and alter the sample structures. So in this way, the electron doses should be not very low and should not be very high, that it destroys the sample. So all these things need to be kept in mind while working with the electron microscopy. So now there are certain strategies to overcome these challenges, such as cooling the biological materials or using negative staining techniques. So there are two ways with which we can protect a biological molecule without collapsing its structure. So first thing is we can go for negative staining, and second step is that we can either go for the cooling of this biological materials, which ensures the minimal electron scattering by using, and to ensure that there is minimal electron scattering, so for that reason we need to use the thin samples, which is ideally a single layer of a particles. So now we will talk about that how the samples are prepared in cryo-EM so that we can deal with all the challenges which we have discussed so far. So single particle imaging, generally in cryo-EM we have thousands of identical particles and we image them from different angles, different directions. So we take 2D picture of one single particle from all the directions, and ultimately we computationally average all those structures to obtain a homogeneous 3D reconstruction of a particle or a biomolecule. So this is what is done in cryo-EM, but there is a possibility that there might happen some compositional variations. So biochemical purifications need to be done so that the sample which we are observing, it should be homogeneous. For example, if we have some contaminations or if we have mixture of proteins, then our aim should be to get a homogeneous sample for which we can perform various purification methods. And this is one of the example of size exclusion chromatography, or means depending on this, gel filtration chromatography or other techniques you can use to purify your sample. Second thing is conformational variations. So buffer conditions can influence the sample appearance by stabilizing the flexible structures. So if you have a protein which is very flexible, so for that reason you can use certain buffer conditions so that it can stabilize that flexible structure. And second thing which you can do is chemical cross-linking, in which you can use a cross-linker which can cross-link the different units of a protein so that it remains stabilized. It does not move much, so that a proper structure can be visualized. But the limitation of this chemical cross-linking is that it can trap the complex in a non-native or inactive form. So in that scenario, even if you get the structure, but if it is in non-native form, then there is no purpose of observing that structure. So then what we can use is the ligand induced stabilization. So if, for example, you are observing an enzyme which has, or a protein which has a ligand to which it binds, then in that case we can use the ligand as a stabilizer. So as you can see here, that stabilizer, what it will do, it will stabilize its structure in one particular orientation, and it will does not allow it to move very flexibly, but still maintaining a proper working structure or a native structure of that particular protein. So for that reason, these are the stabilizations which we can perform. So this was all about the sample preparation. Now comes grid preparation. So we have seen that in cryo-EM we have a grid on which we will put our sample. So there is a requirement of vacuum environment. Biological specimens must be fixed in a native state to prevent dehydration in the vacuum, because when a particle will be in the vacuum, then it might get dehydrated. So to prevent it, we need to fix that structure. Sample thickness should be as thin as possible in case of transmission electron microscopy, because electron beam has to transmit through that specimen Then comes EM grid composition, because ultimately we will put our sample into this grid. So traditionally 3.05 millimeter metal mesh is mostly copper, but gold is preferred because copper can lead to copper poisoning or toxicity. So gold is generally preferred as a grid, and it has the mesh. So mesh means this particular structure is known as mesh. Like a filter, it acts as a filter which can hold your sample, and sample is generally put in this carbon film. So you will keep this carbon film on the gold grid which has these mesh. So if the mesh are more, then it can hold the carbon film or sample very properly. But if it is, there are too many mesh, so in that case it will not allow the proper electron beams to pass through it. So support film is a film which is deposited over this metal mesh. This is known as support film, which is supporting your specimen. So finer mesh provides better support but can obstruct the beams when tilted. So for that reason we need to maintain a balance between this support film and beam accessibility. So the mesh which are generally preferred is 200 mesh grid. In that case it is a balance between the support and beam accessibility. Yeah, so this represents the grid, and in that grid we have a square patch with regular holes in the carbon film. We place this carbon film onto this grid, and this will hold the samples, and ultimately the samples will be present. These are the embedded particles, and the electron beam will pass through these embedded particles and further visualizations will be done. So this is all about the grid preparation. Now comes negative staining, because when we are putting a sample onto this grid, then we want them to be in near native state so that we can prevent dehydration. So for that reason we can either go for negative staining or vitrification. So negative staining, this is the technique in which, why it is called negative, because we are using the acidic conditions in which the stain is acidic in nature and it is negatively charged. So purpose of negative staining is that a rapid method for imaging macromolecular complexes at room temperature. In staining process, the specimens, they are adsorbed onto a thin carbon support film which we have previously seen. We stain them with heavy metal salts such as uranyl acetate, formate. So these are the acids, acetic acid, formic acid. So uranyl acetate, these are the heavy metal salts which are generally used for staining. So the stain forms a cast around the object, enhancing the contrast and increasing the signal to noise ratio. So that is the reason why we are using negative staining. So in this case the example is of nigrosin, which is a negative stain. So as it is negatively charged and we have a bacterial cell which is a specimen, so its cell wall is also negatively charged. Now what will happen, as you add the stain, this outer surface of a bacterial cell will repel this stain. So now my bacteria will remain unstained, whereas entire background, entire media will get stained. So now when I will visualize this specimen, then it will appear bright against the dark background or stained background. So this is the main principle, that why do we use negative staining, because it will further increase the signal to noise ratio and also enhance the contrast of the image. And in this it is also preventing it is also maintaining, allowing it to maintain the proper conformation of the specimen. So this is the principle, that you adsorb the sample onto this grid because you have a carbon support film which is kept onto this grid. Now you will wash it and perform the wash stain cycles. So you will wash it and allow it to blot. So in this case you are allowing your sample to get blotted, to get fixed onto this mesh. So this is done 3 times, and then you add the stain and again allow the stain to get blot, to get fixed to this particular samples. 2 times this again and again staining will be done. Now the image will be collected and data can be analyzed. So there are advantages of this negative staining. First is that it provides resistance to electron damage and prevents the biological materials collapse in the vacuum. So as we are using the dyes which are the heavy metal salts, so it will protect our specimen from the electron beam damage. And second thing, It will also provide the resistance to them and allow us to get better contrast image. But there are certain drawbacks of negative staining, such as potential specimen deformation, because in some instances this can deform our specimen, and limited resolution. So resolution is greater than 20 angstroms, and surface topography restrictions. So in some scenarios we cannot perform staining for certain biomolecules which do not have these. For example, if our cell is not negatively charged, then it will behave differently with the negative stain. So it also depends that what is a biomolecule which you are observing, whether we can perform staining for that or not, whether it has the whether it is unaffected by using staining or it gets affected. So all those things need to be kept in mind while performing the negative staining. So these are some drawbacks of negative staining. And then there are some preferred orientation issues, because continuous carbon support can cause the samples to align in one direction, which can also complicate 3D structure reconstruction. So these are some of the limitations of negative staining. And this is also the representation. This shows that, for example, if this is a specimen and you have used the stain, so this electron opaque stain will not allow the electron beams to pass through it, so they get scattered back, they get reflected back, whereas the other stains, other beams, they can pass through the specimen, relatively electron lucent particle, and image will be formed. So in this way, this shows that how you can increase the contrast, and this is the result. Extracellular vesicles can be are well resolved, they can clearly be distinguished, and they have a proper contrast. So this is the principle of negative staining, and this is also the procedure that how it is done. This is taken from one of the paper in which they have shown that firstly the glow discharge from a foam var and or carbon coated grid to make the film hydrophilic. So firstly film is made hydrophilic so that it can interact with the stain. Incubate the grid on a droplet. So in this way we incubate the grid on a droplet of sample suspension. We blot excess sample away, and then we incubate the grid on a drop of heavy metal stain for 10 seconds so that this grid can take up the stain particles. We allow it to blot. So blot the stain away and dry the grid for 30 minutes before imaging. So blot means, which this term we have also seen a lot of times when we say western blotting, northern blotting, southern blotting. So blotting means when there is some interaction between two different atoms or two different biomolecules, that is known as blotting. So in this case, the uranyl acetate particles, they are interacting with our specimen which is present onto this grid. So now blotting means in this there will be some bonds which will be formed. So for that reason we use we provide 30 minutes so that our stain can blot to our specimen, and then ultimately we can go for performing the imaging. So next thing is vitrification. So because there are some issues with negative staining, because staining dehydrates biological specimens and they also remove them from their native aqueous state. So for that reason we go for vitrification. Cryoimmobilization, this is also known as cryoimmobilization, which preserves the aqueous environment and reduces the radiation damage by freezing samples rapidly, and the speed at which the freezing is done is 10 raised to power 6 degrees centigrade per second. So this is how the temperature is dropped per second. Vitreous ice formation. So if freezing is too slow or warming exceeds, that is negative 137 degrees centigrade, then crystalline ice will form. So as you can see in this case, if there is slow freezing, then the aqueous environment which is present, it will convert to ice crystals which can damage our sample because crystalline ice is formed. But if we use vitrification, which is like fast freezing technique in which we are using this reducing the temperature 10 to the power 6 degrees centigrade per second, so that is known as vitrification. In this case we generally use the cryoprotectants, and when the cryoprotectants are used, then in this case there is no ice crystals which are formed, so our sample get locked in its native state. Yeah, so in case of slow freezing, there is ice contamination which happens, and then there is a scientist named Dubochet who has given this vitrification method. So this is the representation. The sample is transferred to a metal mesh and excess material is removed. So this is the mesh, or this is the grid onto which we put our sample. So now sample is loaded and excess sample is removed. Now the sample forms a thin film across the holes in the mesh when it is shot onto the ethane at about minus 190 degrees centigrades. So this is a cryoprotectant onto which we put our grid containing the sample. So this technique means in which we are dipping the grid with the sample onto the cryoprotectant. So this is known as plunge freezing. So the water vitrifies. Vitrifies means it quickly get freezes without forming the crystals. So it vitrifies around the sample, which then is cooled by the liquid nitrogen during the measurements in the electron microscope. So now this is the representation of one of the grid, how it appears. So now our sample is locked in its native environment. So this is known as vitrification. But there are some limitations with this plunge freezing as well. So this technique is plunge freezing. So now what can happen? That thicker samples which are less than 1 micrometer, such as nuclear or perinuclear regions of eukaryotic cells, they may form crystalline ice which can distort the ultrastructures. So if you have thicker samples, then there is the issue of formation of crystalline ice. So for that reason, we can go for high pressure freezing, in which we apply about 2000 bar pressure while freezing with liquid nitrogen to prevent the crystalline ice formation. So in this case, if you freeze with the ambient pressure, then crystals can still form, and this can disturb the ultrastructures such as these membranes. Whereas if we use the we perform the vitrification at high pressure, then there is no crystals structures which are formed like this case, and in this way the membrane or the ultrastructures, they are also properly maintained. So this is the example. So the advantage of using the high pressure freezing, that it preserves the sample in a near native state, which enables the higher resolution three-dimensional structural studies. But the disadvantage of this is that it requires sectioning or focused ion beam milling for TEM visualizations. So this can also introduce some artifacts, and it is also technically challenging to maintain these conditions. So freeze substitution is alternative. So gradual warming and replacement of water with acetone, allowing staining and resin embedding for room temperature imaging. So this can be done as an alternative to high pressure freezing. So in this case, we generally replace water with acetone. So that can also be done, and then followed by staining and then embedding them for the room temperature imaging can be done. Now here is the comparison between the negative staining versus using vitrification. So when you use negative staining, then it gives you high contrast image because specimen appears light and the background will be dark, so contrast is enhanced. Whereas in case of vitrification, the contrast is the low contrast. Whereas in case of negative staining, there is no special temperature control which is required. Whereas in vitrification, we need to maintain the condition at the cryogenic temperatures because we are performing vitrifications. So in that case, we are doing freezing, quick freezing or fast freezing. In case of negative staining, no radiation damage happens. Whereas in the case of vitrification, there is a possibility of high radiation damage. Particles can get distorted in negative staining, whereas in vitrifications, particles remain undistorted. Image stain the shells around the particle. In this case, images of actual particle because there is no such staining which is taking place. By using negative staining, this is a low resolution method which is 20 to 15 angstrom, whereas vitrification is a higher resolution method which gives resolution up to 15 to 4 angstrom. So that means even if the objects are 4 angstrom apart, then also you can clearly distinguish them. So as I discussed previously also when we were talking about the resolving power, that if resolution is less, that means it has high resolving power. That means it can even distinguish the objects which are very close to each other. So this value need to be less to call it as the better resolution or higher resolution. Negative staining is great choice for initial sample screening, whereas vitrification is best choice for reconstruction. Now we will move to electron source. Till now we have seen the sample preparation, that how the sample is prepared and how grid is used and how that sample is loaded to grid and how we make sure that our sample is in its native state. Next thing comes electron source, because now we will pass the electron beam through our sample so that we can visualize it. So electron gun extracts and accelerates the electrons for imaging. There are two types of electron guns. First is thermionic emission source, so which uses tungsten filament or lanthanum hexaboride crystals, and second type is filled emission gun, which uses fine tungsten tip with zirconium oxide coating. So there is a minor difference between both of these techniques. In case of thermionic emission source, as the name suggests, therm ionic, that means there is some kind of a temperature is involved, heat is involved. So it operates by heating the tungsten filament or lanthanum hexaboride. So tungsten is heated at around 2500 kelvins, and then lanthanum hexaboride, if it is used, then it is heated at 1800 kelvins to emit the electrons. So in this case, as you can see, tungsten filament or this crystal, they both are heated so that they can emit electrons. So the voltage which is required is 80 to 200 kilovolt, and it has lower brightness and coherence, and it is cost effective, and it is generally suitable if you are using the negative staining electron microscopy. Second type of electron gun is field emission source. So field emission, that means it is generating some field. So in this case we use sharp tungsten tip which is covered with the zirconium oxide coating, and it is operated under the high voltage, so which is about 200 to 300 kilovolt, and it operates at room temperature if it is a cold field emission gun, and or at 1800 kelvins if it is a Schottky field emission gun. So this is the types of field emission guns. So if it is Schottky, then it is generally operated at 1800 kelvins depending on the type. It provides high brightness and coherence, and it is ideal for the high resolution electron microscopy, but it is quite expensive. So in this case we are not heating it, but we are providing high voltage. With the help of high voltage, this tungsten tip will release some electrons. Anodes are present, so which will pull these electrons. So electron field is generated in this scenario, which is responsible for moving the electrons or for generating electrons. Whereas in the case of thermionic emission, the heating the filament is responsible for generating the electron beam. Now comes electron detectors. So after the electron beam is generated, depending on the type of electron microscopy which you are using, so depending on that, the electrons will either get diffracted or they get scattered or they transmit through the sample and then ultimately get collected. So depending on that particular thing, we need the electron detectors which can detect the electrons after passing through the sample or specimen. So cryo-EM images, they are inherently noisy due to the low electron doses. So we need to maintain the balance between the electron dose which we use, because if dose is low, then images will be noisy, but if dose is too high, then it can distort or disturb our sample. So in that way we need to keep all these things in mind, that how much dose should be used, which can give us high signal to noise ratio as well as without influencing or disturbing a specimen. So there is a metric which is generally used to check the performance of a signal to noise ratio, which is known as detective quantum efficiency, which is the ratio of signal to noise of the output square divided by the signal to noise ratio of the input and performing its square. So its ratio is known as detective quantum efficiency, that what is the performance of a detector, either it is a good performance or a poor performance. So if we have high DQE value, which means the signal to noise ratio of the output, if it is high, then only DQE will be high, which means the image quality is better. So we use in case of detectors, we use charged coupled device cameras, which are known as CCD detectors, which utilize a phosphor or scintillator to convert electrons which hits into the photons. So it has three different units. First is the phosphor or scintillator. So what will happen is that as the electron beam coming after the specimen is collected by this detector, so electrons will come and hit the scintillator. Scintillator will convert these incoming electrons into photons. Now photons will be created. Photons will be passed from the fiber optic coupling, which will ultimately convert these photons into the electrical impulses. And then these electrical impulses will be collected by these sensors, and which will check that how much electrical impulses are generated, and ultimately image will be formed. So let's see what happens. So phosphor scintillator will convert the electrons into the photons. Photons are transferred by the fiber optic coupling to CCD sensor. So this one is the sensor. CCD chip, it contains a grid of photosensitive elements. So this chip, it has the photosensitive elements where the charge accumulates. So in this case, photon to electron conversion will happen. So charge will be generated and charge transfer process. So now this charge will be transferred. Charge is moved to the readout register. So this one is the readout register, which is reading out this charge. This will amplify it and convert into a digital image. So scintillator layer causes the photon scattering, which reduces the sensitivity. So in this scenario, there is a possibility that photons can get scattered. So in such scenarios, there is a reduction in the sensitivity of image which is formed. But despite these limitations, CCD cameras can achieve sub-nanometer resolution under the optimal imaging conditions, so due to which CCD detectors are mainly preferred. Now comes data collection of protein sample in cryo-EM. So now detectors have collected your electrons which are coming after striking the specimen. Now after detection, how we collect the data? So initial data collection, optimize the protein density and ice thickness for better imaging. So this thing we need to keep in mind, that our sample should be homogeneous and the specimen which is used, it should be very thin so that better images can be produced. Use a mid-range electron microscope to obtain 2D reference free class averages with high resolution secondary structure features, for example alpha helices. So this is just an example of a secondary structure. So we need to make sure that the electron microscope which we use, it should be mid-range. That means the electron beam, its intensity, its dose should not be too low or too high. It should be in mid-range, and then it will make the 2D reference images from different directions. So generally the specimen with which from which we are allowing the electron beam to pass, we generally tilt that sample in different angles. We collect its picture from different directions, so that is known as 2D reference. Or you can say we take the pictures from different directions, different 2D images are captured. Now out of these 2D images, we classify them, that which images are close to each other, which images are taken from the same direction. So we generally average them and take the best out of them. We average them so that there are not any noise which is captured. So to make sure, we perform averaging, and after performing the averaging, different 2D main classes are formed, and from those classes we make a 3D classification. We generate a 3D picture of that particular specimen, and ultimately refinement and model building is done. So preliminary data set and 3D reconstruction. So it collects 100 of micrographs with tens of thousands of particles to generate 2D classes. Use a high-end microscope to compute an initial 3D map. So all these things, they are done by using the computer. So computer programs already exist which will perform this classification, and then for particular reconstruction of the 3D pictures or 3D map. So key evaluations is that 2D class assessment, which ensures the distinct particle views with self-consistent dimensions and secondary structure details. So this thing has to be made sure that the particle which you are viewing, it should be distinct. If they are close to each other, then they will be kept in a single class of this 2D image. And then we also make sure that it should have a proper secondary structure detail. Second thing is orientation distribution. So we verify that sufficient angular sampling for isotropic 3D reconstruction. So from each direction, when we are performing averaging, then we make sure that there should be enough samples which are used for averaging one particular unique 2D micrograph. And then comes optimization. So if orientation is inadequate, then we need to adjust the buffers, detergents, plasma conditions, or we need to use some alternative surfaces such as graphene and amorphous carbons. So what can happen in some scenarios, that our protein is too flexible, if it is too much moving, it have multiple alternate conformations, or due to certain staining techniques, what can happen is that your protein does not get into a native form, it is continuously moving, then we do not get a proper well 2D micrograph. So in such scenarios we need to change certain conditions so that we can maintain the proper structure of that particular biomolecule. So for that reason we need to go for optimization, and these are some of the methods which are generally used to optimize the biomolecule. Then comes high resolution data collection. So if initial 3D resolution is reasonable, then we perform the high-end data collection to obtain a high resolution 3D structure. This is a paper in which it was very well explained that how the electron microscopy is generally used in drug discovery, and this is the pipeline of cryo-EM structure determination which we have discussed. So this is one by one explained. So first of all, homogeneous and highly pure protein sample is applied to cryo-EM grids. Now after it is applied to grids, the sample is rapidly frozen. Vitrification is done in liquid ethane in a thin layer of vitreous ice. Now images are collected as movies on a transmission electron microscope, and these movie frames, they are aligned to reduce the effect of drift. So which means that the motion which is happening, so that motion effect is removed by using the motion correction. So in motion correction we remove the flexibility, or we remove the noise which is coming by performing that averaging and all those things, to make sure that the one proper 2D micrograph which we obtain, that should be properly well resolved. So for that we remove this motion which is happening, so that is known as motion correction. Now particles are picked from each micrograph with those representing the same view grouped together to increase the signal to noise ratio, which is known as 2D classification. So in particle picking, the two particles which appears same, they are classified as a single class. So in this way we perform 2D classification of thousands of pictures which are collected. Now after 2D classification is done, so these 2D classes, they are computationally aligned to generate a 3D map. And now 3D classification, they can identify different conformational states of a protein. So later on refinement is performed and then model building is done. So this is the pipeline, that how the cryo-EM structure is determined. Now there are certain advantages as well as challenges. So advantages is that the resolution which we obtain from these cryo-EM structures, they have enabled the visualization of small molecules, lipids and antibodies which are bound to proteins. So in this scenario we can perform drug discovery. So because if we can design small molecules which might act as drugs, and then we can see that how these drugs interact with the proteins. For example, if a protein, in the later slides we'll see an example, but if my protein is involved in some kind of biochemical pathway which increases when it binds to some other ligand, then it increases the signaling and leads to cancer. So in such scenarios what I can do, I can design a drug which can compete with that ligand and it can bind to my protein. So in such scenario it can inhibit the process which was taking place previously. So with the help of electron microscopy I can see that whether my drug will be interacting with the protein. If yes, then either it is destabilizing my protein. So all those studies can be done with the help of cryo-EM, and wide range of systems can be studied such as membrane proteins, viruses, ribosomes. So these small proteins or biomolecules can be studied with the help of electron microscopy. We can also check the different conformational states as we have seen here. So different conformations can also be studied. Smaller amounts of purified proteins is needed in the EM, as compared to X-ray crystallography or NMR, in which we need the high amount of purified protein. And then boundaries are continuously being pushed because with the advancement which is taking place, so resolution is improving day by day for the cryo-EM, and also the size of the protein which we can observe using cryo-EM, so that is also increasing. Yeah, so with this we can say that the cryo-EM has multiple advantages, but still there are certain certain challenges which are present, such as the time which it takes to determine the structure is ranging from days to weeks, so which is a quite long time, and it is not currently used to screen the large compounds or fragment libraries, because we will struggle to identify the compounds with weak binding affinities. So these are some of the challenges. If you are interested, you can go through this paper. Here is the link. You can click on this link and study this. And this is 3 different proteins which they have studied. One is the LMNG detergent, and there is another protein which is interacting with the lipids, and this one is interacting with nanobodies. So with this they have seen that how the structure changes, how the conformation changes when it interacts with the small molecules. Now comes testing the specimen, testing for the specimen movement during or after the data collection. So what is the importance of motion correction, which we have seen here, that motion correction is done. So it verifies that the dataset quality by checking specimen preparation and illuminization to minimize the particle motion. It uses the motion correction programs such as Motion Core for per micrograph adjustments, micrograph collection and motion compensation. So micrographs are collected as movies split into frames, and algorithms they track that specimen movement over time. So specimen here, for example, is a protein. Now protein can move in different orientations. So there is the program known as Motion Core, which makes sure that how in each frame the particular specimen is moving. So it realigns each frame to correct the motion related information loss. So in this case, as you can see, there is no drift which is taking place, whereas in this case there is motion taking place. So these are not well resolved concentric circles which are formed. So detecting stage drift or specimen motion. So use real-time fast Fourier transforms to identify the stage drift in micrographs. So generally, fast Fourier transforms are used to identify that whether there is motion taking place in micrographs or not. And examples are if there is no stage drift, and this one is when there is 10 angstrom per second drift due to temperature differences or temperature changes. Now comparison of support materials. So gold support versus amorphous carbon on gold, which is known as quantifoil. So gold support versus this amorphous carbon on gold. So we can see that this is the displacement, which tells you how much motion or how much drift is taking place. So if you use gold support, then this displacement is very low, which is less than 1.5 angstroms. So this, which is less than 1 pixel per image. So that's why gold support is much favorable. Whereas if we talk about the quantifoil, which is amorphous carbon on gold, so it has higher movement under the same condition. So this also tells you that by changing the grid, so by selecting different grid material, you can even decide that whether the motion will take place or not. So that's why gold grid is much more favorable in case of electron microscopy. Yeah, so now we'll look into the data processing, single particle analysis. So first of all, image acquisition. So electron beam captures the randomly oriented macromolecular complexes in vitrified solution. Micrographs contain multiple projections of identical particles. So as we know that a single specimen electron beam will pass, and from different directions the image is collected. So which will produce the thousands of snapshots or 1000 micrographs. Now from all those micrographs, which are the random orientations of that particular particle, so from all those micrographs we will pick up the particles. That is known as particle picking. So this is particle picking. We will see which particles can classify or cluster into a single class. So in this way different orientations are taken. Only unique orientations will make the class representative, and all those similar orientation they will be put into a single class. Now within that, for example, here you can see that these are the different classes. In this, particle picking is done, different orientation is captured, and then classification is done. Now from these classes, each of them, they will be averaged to take one representative from each class. So after averaging, one representative is taken from each class. Now we can also perform the alignment to find some new particles, so that averaging can be done better, so that we can increase the signal to noise ratio, and ultimately we can later on go for 3D reconstruction from this 2D images to enhance the resolution. And then computational algorithms reconstruct a high dimensional 3D model which is less than 3 angstrom. So this is all, and this we have also discussed in the previous paper when we were talking about the different steps of the data processing. And yeah, so the paper which I was previously talking, that how the drug discovery is working. So this is the paper in which they have performed the structural identification of R/12 domain of R/R1 protein, which is this domain work as a ADP sensor and the target of malignant hypothermia therapeutic dantrolene. So in this, they have shown that how using cryo-electron microscopy, they have seen the structure of this particular protein, which is R/R1, which bound to dantrolene, and the MH trigger agent, which is 4-chloroamcresol. So MH is the malignant hyperthermia, which is a condition, which is a life-threatening condition, which is triggered by the volatile anesthetics, which activates the pathogenic R/R1 mutants. So in this case, they have seen that this dantrolene, which when binds stacked with ATP or ADP in the R/12 domain. So this one is the domain of my protein R/R1, to which this dantrolene binds along with ATP and ADP, and it induces a conformational change in this domain, which is allosterically coupled to pore closure. So due to close of this pore, there is change in the conformation, which will bring about some changes in the functionality, and which ultimately leads to the malignant hypothermia. So this shows that with the cryo EM, with the help of electron microscopy, we can see that at which domain of the protein my ligand is binding. So in this case, let's take dantrolene as a ligand, which is binding to R/12 domain. So this becomes possible only with the help of cryo EM, that they were able to see that this ligand was binding to R/12 domain, and because in cryo EM we can take different conformations of the protein. So in this, they were also able to trace that by the binding of this ligand to the R/12 domain of my protein, there happens a pore closure. That means conformation of the protein changes as soon as it binds to the ligand. So which shows that how what is the mechanism of the origin of a particular disease and how we can treat it. So if you are interested, you can go through this paper in which they have explained in very detail that how we can use cryo EM for all these studies. And this is the workflow of the cryo EM, so I won't go into its detail. It's quite complicated, but this we have discussed in brief in few slides which we have discussed till now. Yeah, so now let's move to particle orientation. So in data processing, as I have told you, that what we are doing is we are particle picking. We are performing particle picking. So how this particle picking is done, how different images are formed. So in that case, there is different orientations in which the pictures are collected. So particle orientation is defined using the Euler angles before reconstruction. So Euler angles, how we define Euler angles? So first method is we use latitudes, longitudes and rotation to describe the orientation. So this is my protein. I will have 3 coordinates, because in 3 dimensions we have 3 coordinates, x, y and z. So latitudes and these longitudes, so they can be defined, and rotation angles. So that is the first method with which we can define the Euler angles. Second method is that we start with the z-axis as a reference. We rotate this particle. So for example, here we have a particle, so we rotate it around the z-axis and then see that how the x and y-axis changes. So that is the second method with which we can see the Euler angle. Third method is that we fix one of the axis, such as if we fix z-axis and we rotate the particle around z-axis, around x-axis, then different z and y values we can obtain. So in this way, new z-axis location is determined after rotation around the x-axis. So these are three different methods in which we can calculate these angles, which are known as Euler angles, which tells us that in which orientation our protein is currently. So different orientation pictures are taken, and then all these pictures which have been collected, they are helping us to reconstruct a 3D picture. So if a smaller Euler angles and multiple such pictures are taken, then we will get a proper, or you can say well resolved 3D image for our specimen. So 3D reconstruction in Cryo-EM. So our goal is to convert the 2D projections into a 3D volume for structural visualization. So here you can see that each time by 30 degrees the specimen is oriented, at each time its orientation is changed by 30 degrees and its picture is taken. So these are different 2D projections. These are the observed images, and then this is their calculated 2D Fourier transform pictures. Ultimately, 2D Fourier transforms are central sections of 3D Fourier transformations. Inverse Fourier transformation is done to get the 3D density map, and ultimately model is built. So we get a molecular structure. So processes, each 2D projection corresponds to a slice of 3D volume. So if this is my 3D structure, then 2D image will be each slice of it. From each direction, each piece will be combined together to reconstruct this 3D image. So that is what they are saying here. We compute the Fourier transform of all these 2D projections. These 2D Fourier transforms represent the section of this 3D Fourier transform. Inverse Fourier transformation is done to reconstruct a 3D density map, and then model building is done. So ultimately we get a molecular structure of a specimen or a biomolecule which I was studying. Now there is another type of cryo-electron microscopy, which is known as cryo-electron tomography. So CET, which generates 3D reconstruction of biological samples from tilted 2D images, and multiple projections are recorded and computationally stitched together into a 3D structure. So as we have seen in this case, 3D reconstruction in Cryo-EM. So in this case, if I perform tilt series collection, then it is specifically known as cryo-electron tomography, in which same thing is done. We culture the cells, grow them on EM grids, then we perform their plunge freezing to maintain them in their native forms, then production of FIB lamella. So we perform tilt series collection, in which we tilt the sample into different degrees, and from each tilt structure we collect its image. So now images will be collected from different tilt angles. All these images will be taken to reconstruct a 3D image, and then segmentation can be done. So this is known as cryo-electron tomography, and the difference or one more modification is tilt series collection to cryo-EM. Now image enhancement. So what it does is that it due to random variation in brightness or color from the sensor, there might be some noise. So cryo-EM image is typically noisy with low contrast. So we need to perform the smoothing and contrast enhancement of those images, which can be done using this tilt series collection. So if more and more tilt angles will be taken into consideration, then it will give us smooth image, or in which the contrast will be high. So this is a kind of enhancement technique. And if we see the progression of this cryo-EM, so its resolution has improved throughout years. So before 2013, the images were of low resolution, which were about 4 nanometers or even worse. But now we can go for the atomic level resolution, which is about 0.36 nanometers or 1.8 angstroms. So in this example, you can see this is a glutamate dehydrogenase structure, which shows resolution before 2013, and now the resolution is very high, which means the structure gets well resolved. So this shows that there have been a tremendous advancement in the cryo-EM technique, and it has shown the advantages over X-ray crystallography, because it captures the protein in multiple orientations or multiple conformations without requiring the crystallization. It can also analyze the flash frozen proteins in different functional states. So this is the example of HIV envelope that at 2003 its resolution was greater than 4 nanometers. In 2006 it reaches to 3 nanometer. In 2008 resolution was 2 nanometer, and 2017 it was 0.36 nanometers. So as you can see, the value of resolution has increased. So resolving power has increased, although this means you can see that if two objects are only 0.36 nanometer apart, then also we can resolve them. So this shows the advancement of cryo-EM techniques. And as we have seen that it can also capture the protein in multiple conformations. So this is one of the example in which VATPase protein, its pictures have been, it's about 100,000 images were used to create a molecular movie, and this is a rotor shaped enzyme. So in the next slide I'll show you that how this movie is collected. So with the help of this molecular movie, you can even see that how one particular protein changes its conformations and how many conformations it can adopt. So this is the example of bacterial A/V ATPase synthase. It has two different modes. One is ion pump mode, another one is ATP synthesis mode. So in this 10,000 pictures are collected and then they have placed on each other to construct this movie. So now in this it is showing the movement of this part of the protein, which is the rotor. So this is a C ring, and this C ring can either move in anti-clockwise direction or clockwise direction. So if it is moving in clockwise direction, this is known as ion pump mode. If it moves anti-clockwise, then this is the ATP synthesis mode. So in this case you can see this is moving clockwise, so this is currently in the ion pump mode. So this shows you that how with the help of cryo-EM we can even see that how the conformations changes with time and with binding to other ligands or molecules. Now after getting the proper image from cryo-EM, we can submit it to protein data bank. So till now we have seen protein data bank multiple times. So this is a database for 3D structural data of large biological molecules, and sources of data in this PDB are the X-ray crystallography, NMR and cryo-EM, and it is submitted by the structural biologists worldwide. So how data submission is done? So we have to submit the data, and before PDB accepts it, there is a validation protocol. So it validates the structures which we submit. There are certain metrics on which it operates. So after the validation is successfully done, then this structure will be available to all worldwide. So significance of this protein data bank, that this is the only repository for experimentally solved 3D biological macromolecular structures, and it is also freely accessible online. Yeah, so this is the home page of PDB, and for the data extraction there are different modes. So first one is PDB extract, which extract and harvest the data in PDB-X MMSIF format. Then comes SF tools, which converts the structure factor files among different formats. Then ligand expo, in which you can search the chemical component dictionary for ligand IDs. Then is point suits, so generates the symmetry records for macromolecular assemblies. Then comes validation. So in case of validation, it assesses the structural quality by comparing the model coordinates with the experimental data. It also highlights the concerns using metrics such as R-free, clash score and outliers. So there are different such metrics on which the validation is done, and it is checked that whether these outliers are low or high. So depending on this values, it is decided that whether this PDB structure which you have, whether this structure which you have submitted, whether the PDB will accept it or you need to perform certain refinements. So it highlights the concerns using these metrics, and then it provides a summary comparison to help users select appropriate structural models. So because we know in PDB, for each protein, we can have different structures provided by different scientists. So what we can do is that for each structure, so I'll show you an example. For example, I wanted to study some protein which is this one, which is a structural protein in Homo sapiens. So I want to study this protein. So this 8QTB is a PDB ID of this protein. This structure has been submitted by somebody who has worked on this protein. So there could be multiple people. So in this case, deposition author is Trilio Moyo S and Hansen GC. So these people, they have submitted this three-dimensional structure. Maybe some other people have also worked on this particular protein. They have also submitted the structure. So they might have some different PDB IDs. So what I can do, I can take all these PDB IDs and I can compare. I can see the validation reports. So wherever I'll find that this validation report is good, so I can go for it. So this is what is the purpose of this validation report. You can see that how well the protein, what is the resolution of this protein structure which has been uploaded. And you can also go for full report or 3D report, which will further give you more details onto this particular structure. So this method is electron microscopy. Maybe you find another structure which is submitted by NMR or maybe by X-ray crystallography. So depending on you that what you require. So based on that, we can select that particular method and that particular structure. So in PDB, we have different views. So first one is structural view. So in structure view, it provides you the graphical summary of biological and structural protein features of PDB entities. So as we have seen here. So in this, you can select different views. You can see its structure, sequence annotation, electron density, validation report, ligand interaction. So these are different views. So in case of structure view, we can see its structure, its summary, and also its correspondence with UniProt knowledge base sequence. So UniProt is another database which tells you about the protein sequence. So it tells you about different amino acids, whereas in PDB you get the structure of those amino acids combined together in a protein. Second view is sequence view, which is a protein feature view or a genome view. So this is the sequence view, in which the sequence of protein is given in the form of amino acids and it is broken into a secondary structure, DSSP. So it tells you that which amino acids are giving rise to which secondary structural feature. Either it is a helix or beta strand or a turn, which is represented by this blue line. So in this, this is the legend which you can see. So this is known as sequence view. And you can also download the coordinates and experimental data files from the PDB. So in this, you can download the file from this option. You can either download it in PDB format or other formats which are available. So depending on your requirement, you can go for it. Now if we look at the growth of this PDB, then till 2025 it has shown a tremendous growth. From these many years, there's an exponential growth in the number of structures which have been released annually. So the dark blue represents the number of structures released annually versus total number of entries available. So these are the entry available and these are the structures which are released. So till it's 2025 has just started. So that's why this bar is a little low. But from all these years, the number has been increasing tremendously. And if we look at the different methods which are submitting to PDB, so majorly it is X-ray crystallography followed by NMR. But in the recent years, due to the advancement in the Cryo EM, so this EM has also shown rise in the number of structures which have been submitted to the PDB. So it is now nearly similar to the structure submitted by the X-ray crystallography till 2025. So now it is means there is a decline, because we all know that AlphaFold has come up, which can predict the structures with near experimental accuracies. So now we don't have to wait for so many years to get a well-resolved experimental structure. We can even predict the structure using the AlphaFold. But I want to add one thing, that these experimental structures, they are highly confident and they are better than the predictions which have been made so far. But prediction make our task quite easy and quick. So that's why people are moreover shifting to AlphaFold to predict the structure and get some idea. Then comes electron microscopy database. So after PDB, this is the database which contains the cryo electron microscopy structures which are observed from the electron microscopy. So all those structures are present in EM data resource of macromolecular complexes and cells, ranging with resolution ranging from 2 to 100 angstrom. So the goal of this EM data resource is to establish a data validation methods for structure determination, and it define key indicators of well-determined structures for deposition, and it implement the validation procedures for maps and map-derived models. So if we look at the trends of this cryo-EM data, then there is significant growth in the cryo-EM structures, especially after 2015. And we have also seen that the resolution have also increased, which have also improved the accuracy of this electron microscopy. And key discoveries of this cryo-EM are the bacteriorhodopsin, REC-A hexamer, rhinovirus receptor complex, 70S ribosomes, and acetylcholine receptor. Along with this, SARS-CoV-2, all those structures are now well seen with the help of the cryo-electron microscopy only. So if we look at the growth of the cryo-EM maps in electron microscopy data bank and solve protein structure models in PDB with near atomic EM technology improvement from 2015 to 2024. So this is the paper from where I have taken these graphs. So in this, as you can clearly see that EMDB maps, they have increased in the number of released entries with passing years as compared to PDB electron microscopy models. So this EMDB have a greater structures than the PDB EM models. And if we talk about the resolution, then resolution have also improved in these years. So high resolution means less than 5 angstrom and low resolution maps greater than 5 angstrom. So till these orange bars, that means resolution has improved from 2015 to 2023. Then comes nucleic acid database, NDB. So this serves as a portal for 3D structure information about the nucleic acids. So in PDB we have 3D information about the proteins, whereas in this nucleic acid database we have 3D information about the nucleic acids, and it contains experimentally determined structures of nucleic acids and complex assemblies, and it also allows the users to perform search based on sequence, structure, function annotations, and users can also download, analyze and learn about the nucleic acid structures. So in this case, the nucleic acid structures are given by different methods, X-ray, EM and NMR, and if we compare all these methods from 2012 to 2021, then we can see that how the electron microscopy had done the major contributions. From 2013 it has increased in the number of contributions which it does. Earlier it was only the X-ray crystallography which was the major contributor, but now this EM is also contributing nearly same as that of X-ray crystallography to the nucleic acid structures. And if we see what kind of nucleic acids, then majorly the nucleic acid structures which have been observed by cryo-EM are the protein DNA. So the DNA which give rise to protein. And then there is protein RNA. So means in nucleic acids both RNA and DNA comes. So protein RNAs are the next nucleic acid structures which are majorly observed by using these methods, ultimately followed by other DNA and RNA and hybrid nucleic acids. Yeah, and I also want to tell you that currently this database portal is not working. So I have seen this database commons, which is a repository which generally keeps information of different biological databases. So in case if you're not able to find this database, then you can search it in database commons. So in that it will provide you the URL, that which URL can take you to this nucleic acid database or other databases. Yeah, and in this it provides a little bit of description that what information does this server contains, and when it was founded, when it was last updated, either it is accessible, and in which country this is operated, what kind of data it contains, DNA, what is the object, is it the animal or all the taxa it contains the structure. So in that way you can see different informations and different contact informations, that who has submitted, who is maintaining it. So all those things can be found in database commons. Then comes biological magnetic resonance data bank. So as we have seen in this EMDB EM data bank, similarly in EM data bank we had the structures resolved by the electron microscopy. So in case of biological magnetic resonance data bank, we have the structures which have been identified by the means NMR spectroscopy. So this is the home page of BMRB, which will appear like this, and this is the different tabs with which you can perform different operations. So this BMRB is a repository for NMR spectroscopy data on proteins, peptides, nucleic acids and other biomolecules, and it collects, annotates, archives and disseminates the spectral and quantitative data from NMR investigations, and this data is made publicly available worldwide to support the scientific research. And key goal of this BMRB is to help the scientists analyze the structure, dynamics and chemistry of biological systems. It promotes the biomolecular NMR spectroscopy research and development. So these are different views. You can see data archive, which is the already available data in this BMRB. Then there are certain entries. You can also submit your data and you can look for the entries. And this is dedicated to coronavirus. All the NMR structures of the virus which have been deposited, they will be found under this tab. Then if you want to deposit, you can go to this tab. Then in the middle it will open, whenever you will click on any of these tabs, then in the middle it will show the details. So in deposit, these are the different options which you can explore. In new releases, it will tell you all the newly uploaded structures, and these are some of the other tabs. If you're interested, you can go through this BMRB server and see that how it works. Okay, so till now we have seen everything. So I hope it is clear till now that how the cryo-EM works. So today's session was entirely dedicated to the protein structural determination using cryogenic electron microscopy and different types of EM, and how it is uploaded to different databases and what information each database provides you. So now after completing all these, we'll proceed to these MCQs. We'll discuss them one by one and we'll see what will be the right option. So the first question is, what is the journal representation of a PDB ID where X is any integer and BCD are any alphabet? So option A is XBCD, option B is BCXD, option C is XXBC, and option D is BCDB. So you have to tell that which one is the journal representation of the PDB ID out of them. So I'll give you some time and then we'll discuss the answer. Okay, so now let's see. So the right answer is option A, which is XBCD. So here are some examples of different PDB IDs. So this is how the PDB ID appears like. Now we'll see what is the journal rule. How can you find out that whether the ID is appropriate or not. So now this PDB ID, it is a 4-character identifier which uniquely identifies each entry in the PDB. And these 4 characters, they are generally alphanumeric in nature. That means they can be alphabets or the numbers. So they can contain letters A to Z and numbers 0 to 9. That means these all characters, 4 characters, they are alphabets. And then the another rule is no consecutive numbers. So as we all know that in PDB, the first character is always a number. So in all these examples, you can see first character is always a number. So the rule is that there is no consecutive numbers. That means that no two adjacent characters in the ID can both be numbers. So in all of these options, we can clearly see if X is any integer, then we'll only select those IDs which have integer in their first character. So in this way, B and D options cancel out. Then we are left with A and C. Now based on this rule that no consecutive numbers there should be no consecutive numbers. So in option C, we see there are 2 consecutive numbers, XX. So due to which option C also cancels out. So now option A is only left, which is the right answer. And now according to the PDB, currently it is just a 4-character identifier, but they are planning to extend this ID to 8 characters, in which they'll prefix this ID with PDB in future. But till now, it is a 4-character long ID. So I hope it is clear till now that why the option A is right. Now let's move to question number 2. So second question is, what is the final detector of cryo-electron microscopy? Option A, closed compact device. Option B, closed circuit device. Option C, charged coupled device. And option D is charged compact device. Yeah, so we have discussed this as well. So I'll give you some time to think and then we'll see the answer. Okay, so let's see. So the right answer is charged coupled device. So when we were talking about the detectors in Cryo, then we were discussing that this charged coupled device, so there is a scintillator in this indirect detector. We have a scintillator which contains, when the electrons strike on the scintillator, it converts the electrons into the photons, and then those photons will move to the CCD, this which is known as these cells. So in them, whenever the photons will come, they will convert to the electric signal, and then there will be a movement of this charge, and ultimately this detector will convert the charge to the voltage and image will be seen. And this is how the CCD detector actually looks like. So this is like a very small chip, and these CCD cameras are traditionally used in Cryo to detect the electrons and convert them into the digital images. And in modern, there is a direct electron detectors. So instead of these CCD, we have shifted to use direct electron detectors because of their higher resolution and sensitivity. So in this, as I have shown, these are small micro lenses, and these are the light collecting micro lenses which focuses the light from the lens onto individual photodiode cells. These are the photodiode cells. This is the color filter. So after the electron beam will pass through it, the electrons will pass through it, they will pass through the color filter which divide the light into RGB or CMY color components. Then they will enter the photodiodes. So these photodiodes, they receive the light. A photoelectric conversion produces electric charges, that means electrons. So in all of them, scintillator is converting electron to photons, and those light photons will pass through this micro lens and color filter, and they will ultimately reach the photodiode, which will convert that light into the electrical charges or electrons, and then these electrons are sent in vertical and horizontal directions. So vertically and horizontally these electrons will move in this, and the amount sent is determined by the intensity of light received by each pixel. And next, in the CCD output layer, the accumulated electrons are converted to voltage, which creates each pixel's image output. So each pixel is a single unit cell. So in this way, this is the detail about charged coupled device. You don't have to go through it, but this is just an overview that how it works. So now let's move to next question. So next question is, 3D images can be visualized by which of the following techniques? Option A, scanning electron microscope. Option B, compound microscope. Option C, dark field microscope. And option D is magnifying glass. So you have to tell among which all these techniques which can allow us to see the 3D images. Okay, so now let's see what is the right answer. So the right answer is option A, which is the scanning electron microscope. So this is the representation of scanning electron microscope versus transmission electron microscope. So we have seen both of them. In scanning electron microscope, it gives you 3D image, and it is mostly scanning the surface. So it tells you about the surface of the specimen, whereas transmission electron microscopy is also the category of electron microscope which gives you the internal picture of the cell or specimen. So that's why in this correct answer is option A, scanning electron microscope. Whereas if we talk about option B, which was the compound microscope, so they generally produce a 2D image using the multiple lenses for magnification. So in this case, transmission electron microscope also gives you a 2D image. Then dark field microscope, it enhances the contrast for transparent specimens but does not provide true 3D imaging. So in this also, magnifying glass, this is also a simple lens which only provides the 2D magnification of a specimen and not a 3D image. So let's move to our next question. So the question is, the PDB provides information on protein structures determined by option A, X-ray crystallography, option B, NMR spectroscopy, option C, electron microscopy, and option D is all of the above. So now you have to tell me that PDB, it provides you structure from which of these methods. So I'll give you some time to think. This is quite easy. Then we'll discuss. Okay, so in this case also, the right answer is option D, which is all the above. So we have already seen that in PDB we use all these methods, and we also use the combination of multiple other methods, in which the major contributor was X-ray followed by NMR, and currently this electron microscopy has also shown an improvement in its resolution, so it is now also behaving or contributing with similar pace as that of X-ray crystallography to the three-dimensional protein structures. Yeah, so this is about the different methods. Now let's move to next question. So next question is, the whole virus can be best viewed in option A, fluorescence microscopy, option B, scanning electron microscopy, option C, cryo electron microscope, and option D, confocal microscope. Okay, so now you have to think a little and then tell the right answer. Okay, so now let's see the right answer. So the right answer is option C, which is cryo-electron microscope. Now let's look one by one into each option and then we'll discuss. So the first option was fluorescence microscopy. So it is used to visualize the specific viral components using the fluorescent dyes, but not the whole virus in detail. So this is quite obvious that fluorescent microscopy cannot allow us to visualize the whole virus. We can view some of its components. Then if we talk about scanning electron microscope, then you might feel confused that why we are not using SEM. So it provides a 3D surface view of viruses, but it lacks the internal structure details. And another thing is that SEM is a type of electron microscopy. So in this it is not clear that which technique is generally used. Either it is using negative staining or which method it is using. So in such case we are not sure whether we can select SEM or not. Because in SEM, if we use negative staining, then we will not be able to observe the whole virus. So if we are using vitrification and then we are observing the sample, then SEM can work. But if we talk about cryo-electron microscope, then in this scenario, in this it's very clear that the technique which we are using, in that we are performing vitrification. So that's why this option is right. And this is best for viewing the whole virus in near native state at high resolution. Then if we talk about option D, which was confocal microscope, so it is mainly used for optical sectioning of fluorescently labeled samples, and it is not good for the whole virus imaging. So this is a representation of the Melbourne virus, and in this is the structure of this whole structure of the virus, and this is taken from a cryo-electron microscope. This image was taken, so which shows the spatial distribution of these LDBs and the membrane and this capsid. So all these structures, whole virus, can be viewed with the help of cryogenic electron microscopy. This is from the, if the cryogenic technique is used from the SEM, and this is from the TEM. So SEM TEM both are the types of electron microscopy, and cryo is the technique, that with which technique you are observing the sample. Okay, so now let's move to next question. So next question is, which of the following terms is used for the rapid freezing of a sample at a rate of 10 raised to power 6 degrees per second, so that the water in and around the specimen is fixed in a vitreous state? So option A is negative staining, option B is freeze etching, option C is ultra thin sectioning, and option D is vitrification. Yeah, so this also we have discussed. So now you have to tell me which is the right option. And just excuse me for this. So this is 10 raised to power 6 degrees centigrade per second. Okay, so take some time and then we'll discuss the right answer. Okay, now let's see what is the right answer. So in this case, the right answer is our option D. Yeah, so this is corrected in this case. So the right answer is vitrification, because in negative staining we do not do any freezing, and freeze etching is also a different technique. Ultra thin sectioning is just taking a thin section of a specimen, which is generally used in TEM, so that electron beam can properly pass through the specimen, whereas vitrification is the right answer. So it is the process of rapid freezing, and freezing rate is 10 raised to power 6 degrees centigrade per second. And why we adopt it? To prevent the ice crystal formation, because we have seen that if there is slow freezing, then the solvent environment or the aqueous environment outside the biomolecule of the protein, they can form ice crystals, and these crystals can damage the structure of a biomolecule. So that's why we need to go for rapid freezing, which will keep the water in an amorphous or vitreous state. And this technique is crucial in EM to prevent the biological samples, or to preserve the biological samples in their near native state. And this is the technique, so Durochet's vitrification method, in which the sample is taken on a grid, and now this grid is put into a cryoprotectant such as ethane, which is at negative 196 degrees. So this is known as plunge freezing. Now after plunge freezing, this grid is obtained out, and now in this grid the sample will be frozen in the near native state. So that's why this is the right answer. Now let's move to our next question. So next question is, the internal structure of bacteria can be best viewed using which of the following techniques? Option A, compound microscope, option B, SEM, option C, TEM, and option D is bright field microscope. So take some time and then we'll discuss the right answer. Okay, so now let's see. So the right answer is TEM, because we have already seen that in compound microscope we can view the internal structure, but that will be not as well resolved as we can see through TEM. And if we talk about SEM, then it only tells us about the external structure, external surface of an organism such as viruses, bacteria. But if we talk about TEM, then it is transmission electron microscope, with the help of which we can see internal structure of bacteria with better resolution. And if we talk about bright field microscope, then bright field and compound microscope, although they can tell us the bacteria, but its internal structure, by looking to at that much resolution, is not provided by both of these lenses. So the preferred answer is option C, which is TEM. And in this I have provided a difference table of SEM and TEM. So this is very important, that you must understand the difference between both of them, So when we say SEM, it is a scanning electron microscopic technique, and it is a microscopic technique which uses a focused electron beam to scan the surface of a sample. That's why it has got its name, scanning electron microscopy. And if we talk about TEM, then it is transmission electron microscopy, and it is a technique which transmits a beam of electron through the ultra thin section of a sample. In SEM, it scans the surface of a specimen with a focused beam of electron to generate images. In TEM, it transmits electrons through a thin specimen to create an image. So in this case, you can reconstruct a 3D image, but in this way, you can only get an image after getting transmitted through the specimen. In SEM, it typically requires coating the sample with a conductive material to prevent charging, because when you are passing the electron beam through the sample on its surface, then it can lead to charging. So for that reason, we need to coat the sample with some conductive material to prevent charging. And in TEM, it involves the thinning of the sample to provide electron transparency using technique like ultramicrotomy. So ultramicrotomy is also a technique with which you can cut the specimen into a very thin section. In case of SEM, it offers lower resolution, whereas in TEM, it offers higher resolution. So if among SEM and TEM, if we compare, then TEM has a better resolution. And in SEM, it has a larger depth of field, which allow for the more of the sample to be in focus at once. Whereas in TEM, it has much smaller depth of field due to its transmission nature. And if we talk about SEM, then it is more suited for studying the surface morphology and topography of samples. So if our aim is to just to look at the surface, how surface of different bacteria, viruses appears, then we can go for SEM. And if we talk about TEM, then it is used for examining the internal structures and ultra fine details of specimens. So depending on our desire, depending on our need, we can select different among both of them which technique we want to use. So now let's move to our next question, which is question number 8. So which is the first 3D electron microscopic structure released in the PDB? Option A is myoglobin, option B is ribosome complex, option C is cytochrome complex, and option D is bacteriorhodopsin. Now I'll just ask you to focus on this line, which is first 3D electron microscopy, and do not confuse yourself with the first structure released in PDB. So you only have to tell the first structure which is given by this electron microscopy, which was entered into this PDB. So take some time and then we'll discuss the right answer. Okay, now let's see. So the right answer is option D, which is bacteriorhodopsin. So this I have taken from the PDB. So in PDB only, they have mentioned PDB trends, expanding boundaries of complexity with 3DEM. So 3DEM is the PDB ID. 3DEM represents the 3D electron microscopy. So they have shown that how with the help of 3D electron microscopy we can study complex structures of the biomolecules. So in this structures obtained from this 3-dimensional electron microscopy, they have made publicly available in PDB archive to help further scientific research and education. So first entry which has been delivered by this 3-dimensional EM was in 1991, and this was by the groundbreaking structure of bacteriorhodopsin, and since then more than 1700 3-dimensional electron microscopic structures have been made available in the PDB archive, with more than 100 released from 2015 to 17. So remember one thing, that in 1991 this structure was submitted, and which was of bacteriorhodopsin, which they have mentioned. So this is the PDB ID, which is 1BRD. And in 2017, Jacques Dubochet, Joachim Frank and Richard Henderson were recognized with the Nobel Prize in Chemistry for their contribution for the determination of atomic structures using single particle cryo-electron microscopy, which is currently most prominent and highest resolution of 3D EM techniques. So remember, if we talk about three-dimensional electron microscopy, then among them, the highest resolution is of cryo-electron microscopy, followed by TEM and SEM. So two structures from the Frank and Henderson laboratories are included below. So this one was their first structure, which was bacteriorhodopsin, and this is the second of ribosome and EFG. Yeah, so this is just to show you that 2017 they have got the Nobel Prize. So this shows you that what is the potential which lies in this three-dimensional electron microscopy. Okay, so let's move to our next question. So next question is, what is the maximum resolution of a transmission electron microscope? Option A 5 nanometers, option B 0.5 nanometers, option C 0.05 nanometers, and option D is 0.005 nanometers. So we know that transmission electron microscopy has the best resolution till now which we have studied. And then cryo EM has better than this TEM. So out of these, which one is the maximum resolution of TEM? So take some time and then we'll discuss the right answer. Okay, then let's see. So the right answer is option C, which is 0.05 nanometers. So TEM uses the electron beams with much shorter wavelengths than visible light, which allow them to achieve extremely high resolution, and its range is 0.05 nanometers, which is under optimal conditions, which is also known as 50 picometers. So in this way, for your understanding, I have given you the type of microscopy and their maximum resolution and their range. So in case of light microscopy, the maximum resolution is 200 nanometers and range lies from 200 nanometers to 1000 nanometers. If we talk about fluorescence microscopy, then its maximum resolution is 10 to 20 nanometers, and there are some advancements of fluorescent microscopy with STED and PALM and STORM. So these are some super resolution microscopic techniques which works on the principle of fluorescent microscopy. So they have maximum resolution ranging from 10 to 20 nanometers, and its typical range is 10 to 500 nanometers. Then confocal microscopy, it has 180 nanometers maximum resolution, range from 180 to 500 nanometers. In phase contrast microscopy, the maximum resolution is 200 nanometers and range lies from 200 to 500 nanometers. Scanning electron microscopy, it has a resolution of 1 nanometer and range is from 1 to 10 nanometers, whereas TEM, it has the maximum resolution of 0.05 nanometers and range is from 0.05 to 1 nanometers. Cryo EM, it has a range from 0.1 to 0.2 nanometers and 0.1 to 0.5 nanometer is the range. Atomic force microscopy, it also has 0.1 nanometer of maximum resolution, ranges from 0.1 to 1 nanometers. Then super resolution microscopic techniques such as STED, PALM, STORM, so they have 10 nanometer maximum resolution and typical range is 10 to 50 nanometers. Yeah, so this is for your understanding that the TEM has the best resolution among all these type of microscopic techniques. This brings us to the last question of today's session. So the question is, the power of an objective to separate distinctly two adjacent points is known as option A numerical aperture, option B resolving power, option C focal length, and option D refraction. So this is the most easiest question among all these MCQs which we have discussed so far. So I'll give you some seconds and then we'll discuss the answer. Okay, so next let's see the right answer. So the right answer is option B, that is resolving power. So one by one, let's see into different of these terms. So when we say numerical aperture, so it measures the light gathering ability of a lens, impacting the resolution, but it is not directly defining the resolution or separation of points. When we talk about resolving power, so this is the ability of optical system to distinguish two adjacent points as separate. So this is the correct answer. In this, when you are not able to distinguish among 2 points, then this image is not resolved. If you are distinguishing but not properly distinguishing among 2 points, then this is just resolved. And when you are clearly distinguish these 2 points, then it is a well resolved image. Then focal length, it is the distance between the lens and the focal point which affect the magnification but not the resolution directly. And then last is refraction, which is the bending of light as it passes through different media, influencing image formation but not the separation of points. So the right answer was option B, that is the resolving power. So I hope till now everything is clear. So now I'll wait for some time. If you have any doubts, then you can ask your doubts in the chat box, or you can unmute and ask your questions. And if you don't have any questions now, then you can even ask your questions in the discussion forums, or you can simply comment on the YouTube video wherever you feel any doubt. Okay, then if you do not have any further questions, then I'll end the session and I'll see you in the next session. So till then, good night, goodbye.